In Chapter 8, we're going to take a look at synaptic transmission and neural integration. So a synapse is just the junction between a neuron and another neuron, or between a neuron and a muscle, or between a neuron and a gland. There's two different types of synapses. There's electrical synapses and chemical synapses. Chemical synapses are much more common, and that's what we'll be focusing mostly on in this chapter. In electrical synapses, the two neurons are joined together by gap junctions. So you can see in this picture below, you've got one neuron that is on the top here, and the other neuron on the bottom, and you can see how they're joined together by gap junctions. And ions will flow through the gap junctions from one neuron to the next, and that's how the action potentials are transmitted from one neuron to the next. And the action potentials can flow in both directions, so they could flow from the top neuron to the bottom or from the bottom neuron to the top one. And that's going to be very different from the chemical synapses where there's no gap junctions, and also in chemical synapses, the signals can only be transmitted in one direction. We don't find these used very often in the nervous system. One place that we find electrical synapses would be in the brainstem, in the respiratory center that controls the respiratory rate and rhythm. And then we can also find some electrical synapses in the retina as well. Most of the synapses in the nervous system are chemical synapses, where neurotransmitters are going to be released into the synaptic cleft between the presynaptic neuron and the postsynaptic neuron or cell. And signals are going to be transmitted just in one direction. And there's also a synaptic delay, which just means that there's going to be a little bit of a delay in between the time when the action potential reaches the axon terminal of the presynaptic neuron and when the action potential is then generated in the postsynaptic neuron. And that's because there's some time that it takes for the calcium to enter into the axon terminal and then trigger the exocytosis and release of the neurotransmitter into that synaptic cleft. So that's what causes the synaptic delay. So there's three different types of synapses between neurons. There's an axodendritic, axosomatic, and axoaxonic synapse. And they're just named after where the synapses occur between the presynaptic and postsynaptic neuron. So an axodendritic synapse occurs between the axon of the presynaptic neuron and then the dendrite of the postsynaptic neuron. So that one would be an axodendritic synapse. And then an axosomatic synapse occurs between the axon of the presynaptic neuron and then the soma, or the cell body, of the postsynaptic neuron. So this would be an axosomatic synapse. So these axodendritic and axosomatic synapses, we're going to be talking about these most of the time. And these are going to be generating graded potentials in this postsynaptic neuron. And if the graded potential is depolarizing enough and we reach threshold at the axon hillock, then we can generate an action potential which will then travel down the axon. The last type of synapse is called an axoaxonic synapse. So this is between the axon of the presynaptic neuron and then the axon of the postsynaptic neuron. This is a very different type of synapse. It's not going to create a graded potential at all in this postsynaptic neuron. The axoaxonic synapse will just affect how much neurotransmitter is going to be released from this postsynaptic neuron's axon terminal. And so we'll talk about that one a little bit later. So this is a really important picture to understand what's happening at the synapse, all these steps. So the first step, the action potential will travel down the axon and then reach the axon terminal. And in step 2, the action potential will trigger the opening of the voltage-gated calcium channels. Calcium will enter into the axon terminal, and that'll trigger the exocytosis of the neurotransmitter into the synaptic cleft. The neurotransmitter will then diffuse across the synaptic cleft and then bind to receptors that are on the postsynaptic neuron or postsynaptic cell. And then we'll have a response in the postsynaptic neuron or postsynaptic cell. And then the next thing is we need to get rid of the neurotransmitter, because once we've gotten our response in our postsynaptic cell, we don't want to keep responding. So for example, if this postsynaptic cell is a skeletal muscle cell, once we tell it to contract, we don't want to have the muscle keep contracting. So like I wouldn't want to have my fist contracted all day. So I'd want to be able to contract it and then have the muscle relax again. So we need to get rid of the neurotransmitter. And so there's 3 different ways to get rid of the neurotransmitter. So one way is to break down the neurotransmitter with an enzyme. So depending on the neurotransmitter that's used, there might be an enzyme available to do that. So if the enzyme is acetylcholine, the enzyme acetylcholinesterase will chew up acetylcholine and get rid of it. And then the other 2 enzymes, MAO and COMT, they will get rid of amine neurotransmitters and take care of those. Another way to get rid of neurotransmitters is by reuptake. So the neurotransmitter will be pumped back into the axon terminal and then can be either broken down in the axon terminal or be reused. And then the third way to get rid of the neurotransmitter is that some of it will diffuse out of the synaptic cleft and then get taken away by the bloodstream. Let's take a look at the receptors that are on the postsynaptic neuron. So these would either be on the dendrite or the soma of the postsynaptic neuron. And we can have either these ligand-gated channels, which are also called channel-linked receptors. They're also called ionotropic receptors. And that's because these are ligand-gated ion channels that when the ligand binds, the gate will open or close and they'll let ions in or out of the cell. And that's going to be a very fast response because the gate will open and close very quickly. And depending on which ion is going in or out of the cell, that could either be excitatory or inhibitory. So you could have either a depolarizing or hyperpolarizing graded potential that's produced in this postsynaptic neuron. The other type of receptor that could be on this postsynaptic neuron on the dendrite or the soma here could be a G protein linked receptor. These are also called metabotropic receptors. And these are going to be giving a much slower response because it takes longer to activate the G protein, and then the G protein part of it will take off and go do things. And so that's a little bit slower response to get an effect. And depending on what the G protein goes and does, it will either be excitatory or inhibitory. So this is a ligand-gated ion channel. And just showing you with the gate closed here in the middle. And when the ligand, in this case we've got a neurotransmitter binding, that opens the gate and will let the particular ion in or out of the postsynaptic neuron. And then that will create a graded potential, either depolarizing or hyperpolarizing, depending on which type of ion it is that's going in or out of the cell. And this will be a fast response, so they do call these fast ligand-gated ion channels. The other type of receptor on the postsynaptic neuron is a G protein linked receptor. So the neurotransmitter will bind to the G protein linked receptor that activates the G protein. Part of it will take off, and the G protein will do one of two things. So one is it could go open or close an ion channel, and then that will cause a response in the postsynaptic neuron. And the other thing that the G protein could go do is it could go turn an enzyme on or off. And then the enzyme will activate a second messenger system, and the second messenger could produce a cell response by maybe phosphorylating some proteins, or maybe the second messenger will open or close an ion channel itself. So that would cause a response. So by having the G protein be activated and taking off and going and opening and closing an ion channel, or turning the enzyme on and making second messengers, that's a much slower response, and so it's not going to be as quick as the ligand-gated ion channels.